Hello and welcome back to Zoology 141. Today we're going to be looking at Chapter 13, which has to do with the peripheral nervous system and the general senses. Specifically today we're going to be talking about the process of stimulation, sensation, and perception. That is, how do we get a stimulus and convert it into electrical impulses so that that stimulus will be perceived in the central nervous system. So to start out today's lecture, we're going to talk about the peripheral nervous system, and the peripheral nervous system basically encompasses all the neural structures which are outside the central nervous system, that is, outside the brain and spinal cord. So this includes things like our sensory receptors, also the peripheral nerves and associated ganglia, and all the efferent motor endings. So as long as it's not inside the brain or spinal cord, it's technically considered part of the peripheral nervous system. So here's a schematic just showing the different subdivisions of the peripheral nervous system. If you remember last time, the peripheral nervous system is divided into a sensory division and also a motor division. Now the sensory division brings nerve impulses in from the outside of the body into the brain and spinal cord. And so we call it the afferent division because afferent means going towards, that is, going towards the CNS. On the other hand, the motor division is called the efferent division, efferent with an E, and efferent here means that nerve impulses are traveling away from the central nervous system to effectors, such as muscles and glands. Now, the motor subdivision can be further broken down into the somatic motor division and also the autonomic motor division. So the somatic motor division controls voluntary actions in the body, that is, voluntary contraction of skeletal muscle. On the other hand, the autonomic division controls automatic body functions. So this controls things that we do not have voluntary control over, for example, glands and smooth muscle and cardiac muscle. So when your heart rate speeds up when you're excited or scared, that is a result of stimulation of the autonomic nervous system. And the autonomic nervous system is further subdivided into two subdivisions called the sympathetic and the parasympathetic divisions. Now, the parasympathetic and sympathetic divisions are antagonistic divisions. They tend to oppose one another, and we'll talk a lot about these in the next lecture on the autonomic nervous system. So now that we've learned a little bit about the organization of the peripheral nervous system, we're going to focus on sensory receptors. And sensory receptors are structures that help to receive a particular type of stimulus. And the stimulus is basically any type of sensory input, whether it's light or chemicals or heat. This is all different types of stimuli. And so receptors are specialized to convert that stimuli into electrical potentials. Now, once a sensory receptor and its attached sensory nerve generate action potentials, those action potentials will eventually propagate to the central nervous system, that is the spinal cord, and all the way up to the thalamus of the brain, and eventually reach the cerebral cortex. And within the brain, there are two different processes that can occur. The first of these is sensation. Sensation is basically the conscious or subconscious awareness of changes in the environment, that is, awareness of a stimulus. On the other hand, perception is the conscious awareness and interpretation of a stimulus, and this happens exclusively in the cerebral cortex, whereas sensation can happen to some degree within the thalamus. We may become aware of a stimulus, but we can't say exactly where that stimulus is originating from on the body until that sensory information travels all the way up to the cortex, because the cortex is where perception occurs. Now, it's important to realize that not all stimuli are actually perceived. Some stimuli are traveling, let's say, from our blood vessels and from our heart, and they're informing our autonomic nervous system about our blood pressure, about our heart rate, about the osmolality of the blood. So this type of information is transmitted to your brain, but it's not transmitted to the cortex, and so we're not aware of our heart rate, we're not aware of the osmolality of our blood, but these are still things that are being monitored by the hypothalamus and the other unconscious areas of the brain. So as we saw in the last slide, sensation and perception are both processes that occur within the central nervous system, specifically the brain. However, before the brain can analyze a stimulus, we have to be able to encode that stimulus into a pattern of electrical impulses that the brain will understand. And this process is called transduction. Transduction is basically the conversion of a stimulus into a graded potential, and this occurs in the sensory receptor. The other process that has to happen is something called transmission. Transmission involves the actual generation of action potentials or nerve impulses that travel along the sensory axon, reach the spinal cord, and eventually are propagated to the brain. Now, depending on the type of sensory receptor we're talking about, the same cell may be doing both jobs, that is transduction and transmission, whereas in our more complicated sensory receptors, one cell will do the transduction and a separate cell will do the transmission. Now, before we go on to talk about the ways in which sensory receptors are able to transduce and transmit stimulus to the central nervous system, we're going to go on and classify the different types of sensory receptors in the body. And there are several different ways to classify sensory receptors. Probably the oldest and simplest way is to classify them as either general senses or special senses. So general senses are senses that are spread out fairly evenly all over the body. For example, pain, temperature, touch, pressure, and also proprioception are spread out all throughout the body. On the other hand, the special senses are located exclusively in the head, and this includes the sense of taste, smell, vision, hearing, and also balance. So all of the receptors that sense these stimuli are located exclusively in the head, and so we call them the special senses. Now, of course, there are other ways to classify sensory receptors, and three of them are shown here. We can classify the receptors based on their location and also the origin of the stimuli that they detect. We can also classify them based on the type of stimuli they detect. And finally, we can classify the receptors based on their structural complexity. So we're going to go through each of these three categories. So first off, we're going to take a look at the classification of sensory receptors by the location in the body. In starting off, we have something called exteroceptors. And exteroceptors are located towards the outside of the body, and as such, they respond to stimuli arising from the outside of the body. For example, the outside air temperature is sensed by your skin receptors. We also have receptors in our skin for touch and pressure and also pain, and these are all stimuli that are going to be arising from the outside. If you slam your finger in a door, that's an external stimulus and it's going to be detected by an exteroreceptor. So most of the special senses are in fact exteroreceptors, as are many of the general senses. On the other hand, interoceptors detect stimuli that arise from within the body. For example, we'll find interoceptors within the walls of the lungs that'll tell us a little bit about how much we're inflating our lungs when we breathe in or out. And for this reason, these interoceptors are also called visceroreceptors because they're present within the viscera or the internal organs. And these visceroreceptors are sensitive to changes in chemicals, to tissue stretch, and also to temperature changes inside the body. Now, sometimes the interoceptor stimuli are actually consciously perceived, but for the most part, we tend to be unaware of these stimuli that are being sensed and transmitted to the brain because the parts of the brain that receive input from interoceptors are not the conscious areas of the brain. And so areas like the hypothalamus and also brainstem will be receiving information from these interoceptors and making changes in the body to make sure we ensure homeostasis. The big picture here is that most input from interoceptors is not consciously perceived. And finally, our third classification of sensory receptor by location is something called a proprioceptor. A proprioceptor detects stimuli that arise within the connective tissue surrounding joints. They're also found within ligaments and tendons and also within muscles. And basically, they inform the body about the relative orientation of the body's appendages to one another. For example, the proprioceptors can tell us if our arm is extended, if our knee is flexed, and so on. And so we're able to know what our appendages are doing even though we're not looking at them because of the sense of proprioception. So another way to classify sensory receptors is based on the type of stimuli that they detect. For example, mechanoreceptors detect mechanical stimulus, that is things like touch, pressure, and vibration and stretch. And we have a lot of mechanoreceptors within our skin that detect touch, but we also have them within the ear. The mechanoreceptors in the ear help to transduce sound and convert that into action potentials that travel to the auditory cortex located on the temporal lobe. On the other hand, thermoreceptors are sensitive to temperature. For example, if it's cold outside and you start to shiver, that's in part because the thermoreceptors in your skin have been sending nerve impulses to your brain. And of course we have photoreceptors. Photoreceptors are sensitive to light, and the one place in the body that we have photoreceptors would be in the retina of the eye. And it's important to realize that each of these receptor types can only transduce one type of stimulus. There's some exceptions, but photoreceptors generally cannot transduce, for example, mechanical energy very well. If somebody gets punched in the eye, chances are they'll see a bright flash of light, and that's because the retina doesn't know what else to do with this information. It generates some action potentials, and those action potentials travel to the occipital lobe of the brain, and those action potentials are interpreted as light. And that's why you see this bright flash if you ever get hit in the eye. Our next type of sensory receptors are chemoreceptors, and chemoreceptors respond to chemicals. For example, the sense of smell and taste are both based on chemoreception. The food that you eat has molecules in there, and the molecules stimulate certain types of sensory receptors in the taste buds and also in the olfactory epithelium. And that tells us whether or not the food we're eating is good, or perhaps it's spoiled, or maybe even it's poisonous. And so we'll learn more about how chemoreceptors work in the next chapter on the special senses. Now, it's important to realize that we also have interoceptors that monitor chemicals. And these include the chemoreceptors located within the hypothalamus that are monitoring the blood osmolality. It's important to realize that we are not consciously aware of this particular chemoreceptor input. And finally, our last type of sensory receptors are the nociceptors. And nociceptors are sensitive to pain-causing stimuli. For example, if you hit your hand with a hammer, that's going to induce a lot of nociceptors to fire off action potentials, resulting in the perception of pain within the cerebral cortex. Now, nociceptors can also be sensitive to extremes in hot or cold. For example, if you've ever stuck your hand in really, really cold water, it almost feels hot. And that's because while we're also stimulating sensory receptors that have to do with temperature, we're more so stimulating the nociceptors, which transduce that information as pain. And finally, a third way that we can classify sensory receptors is based on their structural complexity. The simplest of the sensory receptors is something that we call a free nerve ending. A free nerve ending is basically just a modified sensory neuron. So take a look at the picture. We have a sensory neuron whose cell body is going to be located in that dorsal root ganglion just outside the spinal cord. Now, it has a process extending away from the spinal cord, and at the end of that process there is a free nerve ending. This free nerve ending is where the process of transduction occurs, and if we have enough transduction occurring, we can eventually have transmission or generation of action potentials. A variation on the free nerve ending is something called an encapsulated nerve ending or an encapsulated dendrite. So here we have the same type of sensory neuron, but if we take a look at the nerve endings of that neuron, they're covered by a special cupula or capsule. And these type of nerve endings tend to be most sensitive to things like pressure and touch. And finally, the last major type of sensory receptor is something that I call a complex receptor. And a complex receptor basically is one that involves a separate sensory neuron and also a separate receptor cell. So here we're dealing with two or more cells that are involved in the processing of that stimulus. So on the left-hand side of the screen we have the receptor cell, which is specialized for transducing a stimulus, and on the right-hand side we have the sensory neuron, whose only job is to generate action potentials. So here the job of stimulus processing is divided between two or more cells. And we tend to find complex receptors within lots of the special senses. For example, the retina of the eye involves several cells that are responsible for transducing that stimulus and eventually converting it into action potentials. So now that we've learned about the different types of sensory receptors, we're going to focus on the way in which these sensory receptors transduce a stimulus and also generate the action potentials or nerve impulses. So let's start out with the simplest of the sensory receptors, which is just basically a modified sensory neuron. So here we see that the modified sensory neuron has free nerve endings. Now, if we stimulate those free nerve endings with an appropriate stimulus, we will form a graded potential in those free nerve endings. And again, this process is called transduction, converting a stimulus into a graded potential. Now, because we have a very simple system consisting of only one cell, the same cell that does transduction will also do transmission. That is, if there's enough of a graded potential in the free nerve endings, an action potential will form and propagate down the axon. In this case, we call the graded potential that forms in this cell a generator potential because it generates action potentials in the afferent sensory neuron. On the other hand, if we have a sensory receptor that's composed of two or more cells, which is a complex receptor, we say that the graded potential occurring in that receptor cell is a receptor potential. And that's because the receptor cell can only form a graded potential. It does the process of transduction, but it can't do the process of transmission. Instead, after forming that receptor potential, the receptor cell will secrete neurotransmitter, which travels across to the afferent sensory neuron. If enough of that neurotransmitter travels across to the sensory neuron, that sensory neuron will then fire off action potentials and transmit those impulses towards the central nervous system. So it's important to realize that most of the general senses tend to be based on modified sensory neurons, which form generator potentials. On the other hand, the majority of these special senses involve separate receptor cells, which are going to form a receptor potential and then activate an afferent sensory neuron. Now, because this chapter deals primarily with the peripheral nervous system and the general senses, we're going to focus mainly on the simplest sensory receptors, that is, the receptors that are based on a modified sensory neuron. Now, the nerve endings for these neurons may be either free or naked, or they might be encapsulated by a connective tissue membrane. Although this might sound very simple, anatomists have described over 12 different types of receptors that consist of either a free nerve ending or an encapsulated nerve ending. A few of these receptors are located on page 486 and 487 of your textbook. So as you might guess, many of these sensory receptors are found within the skin, which is our largest sensory organ. So let's first take a look at the sensory receptors that have unencapsulated or free nerve endings. Now, many of these are located in the epidermis, which is right at the skin surface. These naked nerve endings are unmyelinated and respond primarily to temperature, that is hot and cold, and also to pain. Thus, most of these unencapsulated nerve endings are either thermoreceptors or nociceptors. Now, free nerve endings may also respond to localized inflammation by transducing the sensation of itch, for example, when you're bitten by a mosquito. Now let's take a look at the encapsulated nerve endings. All of these are strict mechanoreceptors, which transduce pressure and touch. Now, the most superficial of these are called the Meissner's corpuscles or tactile corpuscles, and these are located in the upper dermis within the dermal papilla. These are responsible for transducing the sensation of light touch or fine touch and are particularly abundant in the hairless skin, for example on the fingertips. A second type of encapsulated receptor called a lamellar corpuscle or Pacinian corpuscle is located primarily in the deeper areas of the dermis and also the hypodermis. These structures are stimulated by deep, firm pressure, but only respond to the pressure when it is first applied. So now that we've learned about the different types of sensory receptors, we're going to take a look at the process which is necessary to go from stimulation to perception. Remember that stimulation is simply activating a sensory receptor located either within our skin or one of our specialized sensory receptors within our head. Now, in order for stimulation to occur, the stimulus must be a type that the receptor will respond to. In this case, we're pricking somebody's finger with a needle. And so what type of sensory receptors would respond to that? Well, if you said nociceptors, you're correct. And remember, nociceptors are generally going to be a free nerve ending. So let's take a look at the stimulation of this free nerve ending. The process that occurs here is transduction. Remember, transduction is the conversion of that stimulus into a graded potential. Now, if we have enough transduction going on, that graded potential can eventually lead to an action potential. And that's our third process called transmission. So the action potential is transmitted away from the nerve ending up the sensory neuron towards the spinal cord. Now, once that sensory neuron synapses within the spinal cord, it will pass that information on to other neurons which travel up the spinal cord white matter towards the brain. Now, in the process of propagating that message along, a process called modulation may occur. Now, modulation is a way for the central nervous system to either amplify or dampen the magnitude of that sensory information as it passes towards the more conscious areas of the nervous system, that is the cerebral cortex. Now, although both modulation and integration are going on within the spinal cord, we're not consciously aware of this. So it's not until this sensory information reaches the brain that we become consciously aware of the stimulus. And remember, sensation is awareness of a stimulus. And this can happen, in fact, when the sensory information is transmitted to the thalamus. The thalamus, remember, was a relay station to the cerebral cortex. And we may even have a vague awareness of the sensation within the thalamus, but again, we're not able to localize it or interpret it because that is the job of the cerebral cortex. So once the thalamus routes that information to the cortex, we're then able to fully perceive the stimulus. We realize that not only is the stimulus painful, but we can also pinpoint the location of the stimulus on our body. And if the stimulus is really noxious, along the way we'll probably also initiate a spinal reflex to pull that needle away from your hand or pull your hand away from the needle. Now, while we're on the subject of stimulation, sensation, and perception, we need to talk about something called receptor adaptation. Now, sensory receptors can basically ignore stimulus that's constant, because constant stimulus is usually something that's going to be ignored anyway by the central nervous system. And so certain senses, such as touch and smell, adapt very quickly. Basically, after being repeatedly stimulated, these sensory receptors stop firing off action potentials. And without those action potentials, the cerebral cortex is no longer aware of the stimulus. So think of examples of times when your own sensory receptors have adapted. For example, have you ever walked into a room that had a very odd or peculiar smell? Initially, you might walk in that room and even hold your nose, but after 4 or 5 minutes of smelling that particular odor, it tends to fade into the background. And that's because the sensory receptors within the nose adapt very quickly and stop sending information to the cerebral cortex. Another example of receptors that adapt very quickly are the receptors within our skin, that is, some of our pressure and tactile receptors. For example, if you're wearing a watch right now, you're probably not aware that you're wearing it. That is, you're not always aware of the band of the watch around your wrist. It's only when the band of that watch snaps or breaks that you're aware that something has happened. And so once you put that watch on, that stimulus tends to adapt very, very quickly. Now, it's important to realize that not all sensory receptors adapt equally. We already said that smell and vibration and pressure adapt very readily and very quickly. On the other hand, things like proprioception adapt very, very slowly. You can hold your arm out at a right angle to your body and leave it there for 5 or 10 minutes, and the sense of proprioception is still there. Another thing that is not easily adaptable is the sense of pain, or nociception. Nociceptors, unfortunately, don't adapt very readily, and this actually does have a protective function of the body. Because remember, nociceptors tend to be activated when tissue is damaged. For example, think about going to the gym and working out, and all of a sudden you did something to your knee and it hurts a whole lot. Now, chances are that sense of pain is going to be with you for several days, and that's actually a good thing because the sense of pain tells us to stay off that knee until it has time to repair itself. Now, because some receptors adapt more quickly than others, we tend to classify receptors into 2 different categories. Phasic receptors are receptors that adapt very quickly. That is, once they're initially stimulated, they tend to send out a few quick nerve impulses and then they tend to go quiet. And examples of phasic receptors include things like pressure and touch receptors, as well as smell receptors. On the other hand, tonic receptors are receptors that adapt very slowly, if at all. And examples of these receptors would include, again, the pain receptors and also the proprioceptors. Okay, now we're going to move on to talk about thermoceptors. And as the name implies, thermoreceptors detect thermal sensations. That's things like hot and cold. And as it turns out, we have separate hot and cold receptors. The cold receptors tend to be found in the epidermis within the stratum basale. On the other hand, the warm receptors are found deeper down, located within the dermis itself. And these respond to temperatures between 90 and 118 degrees Fahrenheit. Now, both temperature receptors do adapt relatively rapidly, and this is why you can get into a hot tub and initially it just feels like it's the hottest thing in the world, but after 4 or 5 minutes in there, the temperature feels fine. However, even though they do adapt, they do continue to send impulses occasionally to the central nervous system. But even though these thermoreceptors do adapt, they do intermittently send information to the central nervous system so that you are aware of the outside temperature. Now, it's important to realize that pain receptors or nociceptors are going to be initiated when we have temperatures that are really cold or really hot. For example, if we have a temperature that's above 118, 120 degrees Fahrenheit, not only are we going to stimulate the thermoreceptors, but we're also going to stimulate a lot of the nociceptors. Same if the temperature is much below 50. If, say, the temperature is 30 or 20 or something like that, and you stick your hand in some really cold water, chances are that's going to be a very painful stimulus, because at temperatures below 50 degrees, we actually initiate our nociceptors. And speaking of pain receptors or nociceptors, we should remember that pain receptors are unencapsulated nerve endings, and they're found in nearly every tissue of the body. Now, it's important to realize that pain receptors do not adapt very readily. Now, if you want to know how nociceptors detect pain, it really involves several different pathways. For one, injury to tissue releases fatty acids, which are cleaved into arachidonic acid and eventually lead to something called prostaglandins, which cause inflammation. And inflammation causes pain. That is, it stimulates the nociceptors. Other chemicals that are released during tissue damage include potassium and things like kinins, and these also stimulate nociceptors. So now that we're talking about pain, we should mention the fact that there are two different types of pain: fast pain and slow pain. Fast pain or acute pain results from an injury to the outside of the body. And in this case, because the information is being carried to the central nervous system by very thick myelinated axons, we tend to be aware of it very quickly. That is, within about a half a second of the tissue injury. Examples of fast pain would be like dropping a rock on your foot or slamming your fingers in a car door. You're aware of this almost immediately, and again, that's because the nerve impulses are being carried by very thick myelinated axons. On the other hand, slow pain is pain that often originates from the inside of the body. And here the pain impulses are being carried by small diameter fibers which are unmyelinated. And so the information travels relatively slowly from the internal organs up to the cerebral cortex. And so as a result, slow pain tends to develop gradually. It begins a little bit and then increases in intensity over minutes and hours. And so we tend to get more of an aching or throbbing sensation rather than a stabbing pain. Now, when we have pain originating from the inside of the body, for example, the viscera, the area that you feel the pain may not necessarily be the area that's actually injured. And we call this concept referred pain. So referred pain is when we have an injury to one area, but the pain is actually felt in another area. And the reason this happens is that action potentials from internal organs are carried by the same spinal nerve segments that transmit action potentials from more superficial structures. And as a result, oftentimes we get a confusion about where the actual origin of pain is. So this picture just shows a diagram of the different pain fields for our internal organs. For example, you can see the pain field of the heart, which is indicated in blue, and also the pain field of the esophagus, which is indicated in yellow. And what you should notice here is that the green area indicates the area where the pain fields of these two organs overlap. And this is the important part about referred pain, because it can feel like it's coming from one area, but in fact can be coming from another. For example, you've heard of something called heartburn. And heartburn is a pain that usually originates in the lower part of the esophagus or upper part of the stomach. And even though it sounds pretty serious, heartburn is something that's usually not very serious. It's basically caused by a little bit of acid refluxing from the stomach into the esophagus. But because that pain occurs in the area of the chest where the heart is located, people came to call it heartburn. And for that reason, pain that's felt in the chest and the vicinity of the heart is not necessarily cause for alarm. It just depends on what the origin of that pain is. If it's heartburn, it's going to be something that's relatively not serious. On the other hand, if it's angina or pain that's resulting from injury to the heart, that is more of a serious situation. But because these two pain fields overlap broadly, it's sometimes hard to distinguish between heart pain and esophagus pain. For this reason, when people come to the hospital complaining, let's say, of chest pain or heart pain, we often ask them whether that pain is radiating to their shoulders and their arms, because you can see that the blue area that's running down the arm is unique to the heart. That is, esophagus pain never radiates down the arms, and so pain that originates in the chest but shoots down the shoulder and arm is a little bit more diagnostic for indicating somebody is having a myocardial infarction or some other kind of episode with their heart. Now, before we move on from sensory receptors, we need to say a little bit about proprioceptors. Remember, proprioceptors are structures in the body that we find within tendons, muscles, and other tissues that let us know about the relative position of our limbs and joints relative to one another. The proprioceptors can be divided into three categories, and these include muscle spindles, which monitor change in muscle length, tendon organs, which monitor the stretch of tendons and sometimes ligaments, and finally something called joint kinesthetic receptors. These joint kinesthetic receptors are located in the synovial capsule of our synovial joints and they're responsible for informing the cerebral cortex as well as unconscious areas of the brains about the relative stress within a joint cavity. Now your textbook goes into a lot more detail about the 3 different types of proprioceptors, so be sure to read about these in your textbook. Okay, now that we've learned about the process of transduction and transmission that happens within a sensory receptor, we're going to go on to talk about sensory integration. Now, integration, remember, is the process of transmitting that information through the brain and spinal cord and the interpretation, analysis, and filtering that goes on there. And so integration can happen at 3 levels. We have the receptor level, that is the area where transduction is happening. We have the circuit level, that is the transmission of that nerve impulses from the sensory neuron to the spinal cord and eventually the thalamus. And finally, we have the perceptual level. The perceptual level involves the processing of this stimulus within the cortex of the brain. So this slide just shows an overview of the 3 different types of neural integration that go on with sensory information. So let's start down with number 1 at the receptor level. Remember, receptors can be things like free nerve endings or muscle spindles or proprioceptors that are located within joint capsules. Either way, the stimuli that these receptors detect is transduced into a graded potential and eventually transmitted as an action potential to the spinal cord via sensory neuron. We then enter the circuit level, and this is where processing happens within the spinal cord and also the lower areas of the brain. Basically, within the circuit level, the nerve impulses are propagated up the spinal cord to the relay stations of the brain, which include the thalamus and sometimes the pons. Now, once we reach the circuit level, those nerve impulses will be propagated up to the appropriate region of the cerebral cortex, where both sensation and perception occur. Remember that if we're talking about touch, the sensation is first processed when we reach the somatosensory cortex. And that's going to let us know that, oh, okay, I'm touching something. I can't really say what it is because that interpretation doesn't happen until those data move from the primary somatosensory cortex to the somatosensory association area and finally the multimodal association areas. So it's within the association areas that we're actually able to interpret and perceive those stimuli fully. So the big picture here is that in order for a stimulus to reach the brain, it has to travel along a minimum of 3 neurons. So the first of these neurons will be called the first order neuron, and this is a sensory neuron, and this is the one that transmits the nerve impulses from the sensory receptor to the spinal cord. Now within the spinal cord, that first order neuron will synapse with a second order neuron, which will transmit that information to the thalamus, and finally the third order neuron will carry the information from the thalamus to the appropriate region of the cerebral cortex. Now, if we're talking about somatosensory data, it's important to remember that the touch receptors on the body are mapped out onto specific regions of the cerebral cortex, and we call this the somatosensory cortex. So that if I touch my right index finger, my brain has a certain part of the cortex that is devoted to receiving sensory data from that structure, and so we're better able to pinpoint the area of where the stimulus is coming from. So in the past couple slides, I've been trying to drive home the point that sensation can occur within the cortex and also within the thalamus to some extent, but that perception specifically happens within the association areas of the cerebral cortex. But what is perception? Now, we said at the beginning of the lecture that this is conscious awareness and interpretation of a stimulus, but what does that entail? Well, there's 3 or 4 different things that are going on during perception. First of all, we have perceptual detection, which is just basically the ability to detect a stimulus. So this is analogous to sensation. And then we have something called magnitude estimation. We can estimate how strong that stimulus is based on the pattern of nerve impulses that are coming from our sensory receptor. For example, think about stroking your hand lightly with a feather. That's going to initiate nerve impulses that travel up to your somatosensory cortex that are coded to that particular region of your body, the palm of your hand. Now let's imagine that I take the same hand and then I slam it with a hammer. Okay, that's a stimulus that's in the same area, but it's a much different stimulus. And so this will be encoded a lot differently by the pattern of nerve impulses. And so your thalamus is still going to be routing that data to the same exact region, but the data will be perceived much differently because the pattern of nerve impulses is adjusted based on the magnitude of the stimulus. Another aspect of perception, particularly of somatosensory data, is spatial discrimination. Think about your ability to stick your hand in your purse or in your pocket and feel around at all the different things that dwell there.
even though you aren't looking at them, you can discern what something is and what it looks like just based on your sense of touch alone. So we have this ability to do something called spatial discrimination, to figure out how far different parts of this object are apart and get a 3D dimensional image of this in our mind so that we can identify it. Another aspect of perception is our ability to do something called feature abstraction. Now, feature abstraction is a mechanism by which a neuron circuit can be tuned to one feature in preference to others. And this allows us to identify more complex aspects of a stimulus and also divide it into several different types of stimulus properties. And then we have something called quality discrimination. This is the ability to distinguish between several different types of the same sense. For example, when we look at the sense of taste, our brain is able to ferret out all that sensory data coming in and distinguish what is sour, what is sweet, what is bitter, what is umami, based on that pattern of nerve impulses. And again, something that's sour or bitter may make you very unhappy, whereas something that's sweet or savory may make you very happy. So it's all based on our ability to discriminate between these different types of stimuli. And finally, we have pattern recognition. Pattern recognition is the ability to send this data to an association complex or multimodal association complex where we can recognize things that we've seen before. If it's a taste, for example, you can recognize that that particular taste that you're tasting is sushi or it's wasabi. If you're talking about the sense of vision, pattern recognition can help us to recognize faces. And finally, when we're talking about sound, pattern recognition helps us to recognize that this sound is this particular song that we heard on the radio 10 or 15 years ago. So all of these are very important aspects of perception. And remember, perception is the conscious awareness and interpretation of data, and it happens exclusively within the cerebral cortex. Specifically, it happens within the multimodal association areas. So now that we've completed our discussion of sensation and sensory receptors, we're going to switch gears to take a look at some other structures that are part of the peripheral nervous system. And the main parts here include things that are the peripheral nerves. Now, the term nerve is different than a neuron. A neuron is a cell that generates action potentials, and most of these cell bodies, remember, are found within the central nervous system. A nerve, on the other hand, is a very large structure that just contains portions of a neuron. Specifically, a nerve is a cord-like bundle of axons. And unlike neurons, which are very tiny structures, nerves can be very large structures. They can be visible to the naked eye. I'm sure you've been sitting at Kentucky Fried Chickens or somewhere eating fried chicken once, and you bite into that chicken, and then you pull back, and all of a sudden, this gray, greasy thing flops down under your lip. It looks like a blood vessel, but it's not. It's a nerve, and a nerve is just a bundle of axons. And just like muscle tissue, the nerve tissue is divided into regions by connective tissue coverings. And these include endoneurium, perineurium, and epineurium. So here we can see a picture of what a nerve would look like. Remember, the outside of the nerve is covered by a connective tissue called epineurium, and within the nerve, the axons are divided into bundles called fascicles. You probably remember back to the muscular system when we said a fascicle is a bundle of muscle fibers. In the same way, a fascicle is also a bundle of nerve fibers or nerve axons. And so the axons, again, can be myelinated or unmyelinated, and the individual axons may be covered by something called endoneurium, whereas the fascicle itself is covered by perineurium. So just to recap, epineurium on the outside, perineurium surrounding the fascicle, and endoneurium surrounding the axons. So nerves can be classified according to the direction in which they transmit nerve impulses. Sensory nerves transmit impulses from the outside or senses into the central nervous system, whereas motor nerves transmit information from the brain or spinal cord out to the effectors. But these can be seen as sort of like one-way roads or one-way streets that you have in a downtown area, when in truth it's much more common to have a mixed nerve in which we have information going in both directions. So mixed nerves contain both sensory axons and motor axons, and the majority of nerves in the body are in fact mixed nerves. Other structures that are part of the peripheral nervous system include the ganglia. Remember, a ganglion is a collection of neuron cell bodies that's located outside the central nervous system. And there are two places where we have ganglia. One place is within the dorsal root ganglion, which connects to the spinal cord. And remember, the dorsal root ganglion contains the cell bodies of sensory neurons. Another type of ganglion that we'll discuss in the next chapter are the autonomic ganglia. And autonomic ganglia are ganglia that contain the cell bodies of motor neurons of the autonomic nervous system. And we'll talk more about these in the next chapter. Okay, we're going to close out this lecture by talking about the regeneration of nerve fibers within the peripheral nervous system. Now remember, the term fiber here just refers to axons that we find within a nerve. And so it's important to realize that neuron cells themselves cannot regenerate. Once a neuron is destroyed, it's destroyed. However, if we sever an axon, it's sometimes possible to regenerate that axon, at least if it's within the peripheral nervous system. So in the next few slides, we'll talk about the process through which a damaged axon can be regenerated and replaced. So first, let's take a look at what happens when an axon is damaged. So when an axon is cut or crushed, what happens is the axon ends separate. Now, if this injury is actually located very close to the cell body, it could very easily lead to the death of that sensory neuron. However, if it's a long way down the axon, as it usually happens, what will happen is those two ends of the axon will begin to seal themselves off. Now, once those two ends of the severed axon are sealed off, the site distal to the injury will begin to degrade fairly quickly. And this is because it's not receiving any nutrients, and at the same time our macrophages are going to move in and break down any dead or dying cellular material. In the next step, the Schwann cells, whose job is normally to myelinate the axons, will actually help in the process of regeneration by forming something called a regeneration tube. A regeneration tube is basically just a hollow tube which is helping to guide the sprouting axons coming off the proximal part of the neuron to better reach the areas of original contact. And so these rootlets or filaments will move through the regeneration tube and eventually, or hopefully, be able to reach the original area that was innervated by that neuron. Now, it's important to realize that in order for this axon regrowth to happen successfully, the gap between the developing axon and the neural lemma downstream can't be too large. If it's too large, chances are that developing axon will never find the correct tube to go through. And that's because as the tissue heals, other structures will tend to get in the way. And finally, if everything goes successfully, our new axon will bridge the gap and make it into that regeneration tube. Now, that regeneration tube will eventually be reorganized into a new myelin sheath by our Schwann cells. Now, the process of axon regrowth can be relatively slow. It happens at the rate of about 1 to 2 millimeters a day. So if you have somebody that has a severed nerve, let's say in their arm or in their hand, it can take quite some time before these axons can regrow and restore sensation to the injured areas of the body. Now, although we can have regeneration of axons within the peripheral nervous system, we usually don't have regeneration of severed axons in the central nervous system. And that's because even though the central nervous system has myelination just like we did in the PNS, the myelinating cells here called oligodendrocytes actually secrete inhibitory chemicals which prevent the regrowth of the axons. As a result, when somebody has an injury to the spinal cord or brain that severs axons, that injury tends to be permanent. However, scientists and neurologists are studying treatments where we can try to neutralize the growth inhibiting factors secreted by these cells and try to coax the axons to actually regrow themselves. Okay, you've reached the end of the section on the peripheral nervous system in general senses. Just like normal, there will be a short quiz at the end of this presentation. You won't be graded on this quiz, but if you get less than a 70%, I do suggest you go back and rewatch this presentation again and take detailed notes before going on to take this week's Lylema quiz.